Today we'll be extracting the DNA from both the second site sample, Bryn Mawr pond for metabarcoding, and 2 critters from either site for target barcoding. Like in week 3, we'll be following the QIAGEN DNeasy blood and tissue protocol, which requires me to first remove the solution from the second site 1.5 ml tube and avoid touching the filter. To review, I'll be using AL buffer, ethanol, AW1, and AW2 buffers to extract the DNA. Now that my sample has been isolated from the filter and put in a new tube, I'll add the AL buffer and ethanol, making sure I mix thoroughly. After vortexing, I'll be adding AW1 buffer, centrifuging, and adding AW2 and centrifuging again. Once I've added 2 rounds of nuclease-free water to elute the DNA from the spin column, I'll be done, and I put the sample at the instructor's bench to be frozen until the next lab period. One of the 2 critters I selected to target barcode was a spider I collected at the Bryn Mawr pond. I put the spider in an epitube and with a melted 1000 microliter pipette tip, crushed it to break open its exoskeleton. Then it was time to put my target barcoding samples in the 56-degree water bath for an hour. After an hour had passed, I repeated the same procedure that I followed for DNA extraction on the metabarcoding samples for these 2 target barcoding samples. So that includes the AL buffer and ethanol, then centrifuging, adding AW1, centrifuging, AW2, centrifuging again, and finally the elution steps. Once I was done, I added each solution for the samples to an epitube, and it was time to call it a day. On Thursday, we're going to quantify the DNA in each sample using the Nanodrop instrument. The Nanodrop is like a small spectrophotometer where instead of a glass tube, the solution itself is going to be pipetted with 2 microliters onto the reader where the drop is held together through capillary action. Based on the reading, we'll be able to determine the concentration of DNA and based on the quality of light, understand the quality of DNA. The results will yield a curve, and if there's a peak around 260 nanometers, we'll be able to conclude that there is DNA present in our sample. So looking at the duck pond metabarcoding sample, we can see there's DNA present as there is a peak at 260 nanometers. Bryn Mawr pond target barcoding sample, which was a crushed spider, had a very strong DNA presence as there is a large peak at 260 nanometers. During Monday's session, we'll be conducting PCR on our target barcoding sample, and the professors ordered primers based on the ones we indicated that would yield best results. There are 2 samples that didn't have any DNA present. They were the duck pond target barcoding sample and the Bryn Mawr pond metabarcoding sample.